Good morning, everyone. So I think we'll get started now because it's 11 a.m. And welcome everyone to session number 4. So this is a session on Culturomics versus Metagenomics for surveillance. So my name is Carolee Carson, and I'm a veterinarian and an epidemiologist working for the Public Health Agency of Canada. And I'd like to turn it over now to my co-chair to introduce himself.
Good morning to everyone. My name is Iakovos Iakovidis, and I'm in chemical engineering in the background, and I'm coming from the University of Cyprus. Welcome all to this session.
And now it is my great pleasure to introduce our invited speaker, Dr. Amy Pruden. So Amy is the W. Thomas Rice Professor and University Distinguished Professor in the Department of Civil and Environmental Engineering at Virginia Tech. She earned her BS in Biological Sciences in 1997 and her PhD in 2002 in Environmental Science, both at the University of Cincinnati. Her research is focused on understanding the fate of antibiotic resistance genes in aquatic, terrestrial, and airborne environmental systems. And she is currently leading a transdisciplinary PhD training grant funded by the U.S. National Science Foundation focused on translating metagenomic-based surveillance of AMR into policy and practice. She is the recipient of the Presidential Early Career Award in Science and Engineering, the Paul L. Busch Award for Innovation in Water Research, the Recipharm International Environment Award, and is a fellow in the International Water Association. And the title of her presentation is Antibiotic Resistance Genes as Environmental Contaminants, Where Are We Now? Over to
you. Well, thank you very, very much for the kind introduction. It is truly a pleasure to be here. Where is here? Well, it is EDAR7. It is May 27th, 2024. So a time for reflection. How did we get here, and maybe a little bit about where we're going from here? Well, my journey began in the early 2000s. I was an assistant professor at Colorado State University. And rather than explore caves like Jerry talked about yesterday, which are very claustrophobic for somebody like me, fortunately, we had this grand idea to go into the Rocky Mountains. So just imagine the Rocky Mountains. It's pristine, there's snow melt. And so the idea was similar, to see what we could find in terms of antimicrobial resistance genes. So this is a little bit about what we found. So we mapped wastewater treatment plants and animal feeding operations in the watershed, and we found a near perfect correlation with the SAL1 antibiotic resistance gene. And so you might think in this audience, well, of course, but this was a surprising result at the time. And we weren't alone. It does seem that around 2005, 2006, if you search Web of Science for ARG and contaminant, there was basically exponential growth in this research area at that time. And it was really remarkable, the independent validation that we saw in many environments, including Lake Geneva, the Almendares River in Cuba, the Thames River, and polluted river in India, and then also in soils irrigated with wastewater. And so then we got another surprising finding, of course. Okay, there we are. So hats off to Ed Top, who really saw this all coming. He saw the ripple effects of this wave of research and realized we need to get like minds together in a room and wrap our heads around the concept of antibiotic resistance as an environmental problem. And part of what was really cool about this was it wasn't just like-minded people. I mean, many of us had been doing the research independently. It was the first time we met. But he also made sure there were skeptics in the room. So we had veterinarians and doctors and people who work for the pharmaceutical industry. And so I think that made for a much more robust outcome. One of the skeptics was actually Joachim Larsson's own PhD student at the time, Johan Bengtsson Palme, and he put this blog out to start the meeting that ARGs are not contaminants. And so we had a lot of really good discussion, and I think that kind of where we landed, okay, here's the case why they are contaminants. So their anthropogenic origin, as we saw, their spread is undesirable from a human health and animal and plant health standpoint. And we know because of horizontal gene transfer that they transcend their bacterial hosts. And also, targeting ARGs directly avoids the culture bias. But the question, what we choose to target does matter from a policy standpoint. And there's supposed to be some nice pictures of horizontal gene transfer. So one of the main outcomes of EDAR1 was a series of white papers, which I think have been pretty influential in the field. So I hope you all come on Wednesday inspired to take that opportunity at the workshops and contribute yourselves to white papers such as these. The one on the left was one focused on mitigation opportunities. So me as an environmental engineer, we think about, okay, what can we do about it? How can we remove resistance from waste streams? And then the one on the right was focused about risk assessment. And this continues to be a sticking point, because if you want to regulate the contaminant, you really need a risk assessment to get at things like dose response and what is your regulatory limit and of what. Okay, so Johan Bengtsson Palme had instigated this discussion about what is the contaminant, and if we came out of EDAR1 with the white papers and had all these great ideas. And so the EDAR community was forming and making the case that AMR is an environmental problem. And it turned out people were listening. And so within the US, the Water Research Foundation said, wow, we believe you. We're a consortium of water utilities. We want to do something. So what do we do? So then we're scratching our heads like, yeah, that's a good question. So what do you monitor and where? And so I think of it sort of like, have you ever read The Hitchhiker's Guide to the Galaxy by Douglas Adams? It's a good read if you need some entertainment, but kind of the upshot of it is the question's more important than the answer. And so as part of this Water Research Foundation project, we surveyed experts, we had a workshop, we scoured the literature, and where we landed were on these 4 questions. So are you trying to do wastewater-based epidemiology? So looking at ARB and ARG carried in the community. Are you trying to quantify resistance that escapes a wastewater treatment plant and could pose a public health threat? What about removal rates, right? That's great for environmental engineers. We can design better processes. And then the 4th would be, what are those hot spots for evolution and dissemination of AMR? And so this is going to dictate where you monitor and what you monitor. And so if you're interested, we've published several literature surveys and extracted data from the literature. And so this is a paper that Ishi Keenum led on qPCR, looking at targets. And so PCR is still unparalleled in terms of sensitivity and the quantitative capability, but you have to choose a target. And so that's where metagenomics has really been a game changer that I couldn't have imagined. So it's non-targeted, so now you don't have to choose a target. But it's not a panacea. There's still limitations. So there's an inherently high detection limit, and also the expertise required. But then again, it comes back to the question that you're answering. So depending on your question, that dictates how you look at the metagenomic data. And here, Ben Davis led a review that is kind of a modest proposal of what we can do as a community to make our metagenomic data more robust and comparable. And then this is a paper then where we're looking at the culture-based methods. So those are not going away. It's the only way to know you have a viable organism, to confirm a pathogen. But they are a lot of work. Any graduate student will tell you, Ishi will tell you, this is a study from her dissertation where we did culture-based monitoring through wastewater treatment plants and then multiple barrier water reuse treatment. And basically, you know, if you're doing E. coli or even enterococcus, there's nice standard methods for wastewater that you can build on. But we're interested in a lot of other organisms as well, and those tend to be trickier because the media are usually designed for clinical samples, and so they're often very nonspecific. So then, again, you know, what you're targeting and your method is going to depend on your question, and it's going to shape whether you can get at important policy recommendations, right? So what are some practical outcomes that could come from environmental monitoring, right? When we boil it down. So we could inform best practices for prescriptions and antimicrobial use. We can inform public health agencies about an emergent threat or outbreak. We might advise that we should segregate certain wastes like hospital waste before treatment. We can improve treatment processes. We can identify discharge limits. We can maybe inform recreational guidelines. And in the end, maybe prioritize action where it's needed. So let's look at a few examples of some of our recent research. So these data, they might look like metagenomic data, but they're not. This is prescription data that with collaborating with Peter Vickesland, Connor Brown, and Ayala Miley-Moskowitz, our postdocs, we were able to nurture a relationship with our local hospital and convince them to share these kinds of data. And it's quite interesting. One of the burning questions was whether the world went crazy with antibiotic use during the pandemic. So this is kind of before, during, after pandemic. And on the left, oops, I meant to use the thingy. On the left there are inpatient prescriptions, and there's a very slight uptick, that teal-colored bar of inpatient antibiotic use, but on the whole, there's actually been a downward trend in our region, in Virginia, of inpatient antibiotic use. But what you see is during the time of the pandemic, outpatient antibiotic use really plummeted. Basically, people stopped going to the doctor, right? But then you see the cycles going up. So basically, the respiratory illness came back with a vengeance, and here we are, right back where we started. But one thing we weren't expecting, so that these are two communities, and the data are aggregated by the wastewater treatment plant, the sewer shed. The community at the top, there's a lower rate of outpatient antibiotic prescriptions. So we thought, I wonder how that affects the ARGs and the wastewater. So we've teamed up with Li Ching Zhang and computer science and taken a look at the metagenomic data. Now you are looking at metagenomic data. And here what Ayala did is to parse out which ARGs were unique to each wastewater treatment plant and plot those. And I think it stands out that the wastewater treatment plant at the bottom, the one that had higher outpatient prescription rates, also had higher abundances of unique ARGs. And maybe this seems obvious, but it's really hard to show these kinds of examples. First, to get that kind of prescription data, and actually show these kinds of associations. And this really has a lot of potential then. You can imagine then that you could start to provide some guidance on prescription practices and what's less likely to lead to the amplification of resistance. So then, around the same time, Connor and Ayala were busy in the lab with a very cool bench scale experiment where we had more control on what's going on, and they did triplicate reactors simulating wastewater treatment plants. You know, these are complex biological systems, so it's really nice to have those triplicates. And the idea was to feed either 0%, 1%, or 10% of hospital wastewater and see what happens. And so they, at the same time, isolated more than 450 resistant bacterial isolates, did a lot of deep sequencing, both short and long read, and we also had the opportunity to monitor antibiotics. And so Connor got to work digging through the metagenomic data and basically doing hybrid assembly with the long reads and short reads to see where are those ARGs? Are they associated with mobile genetic elements? And so he tapped into the mobile ARG database that he developed and looked at transposable elements, conjugative elements, integrative elements, plasmids, et cetera. So the upshot is good news, and I think we're seeing this across multiple studies at this conference, that a well-functioning activated sludge system largely attenuates mobile ARGs. So that's good news. But I said largely attenuates. There's still some horizontal gene transfer happening. And so what's going on there is probably important to zoom in on then. Now, another somewhat surprising result. So we brought in my collaborator, Kang Sia, who is an environmental chemist, and said, let's look at the antibiotics. And so kind of the point here was the hospital sewage would be loaded with antibiotics, and then we're blending it with our local municipal sewage. Well, the hospital sewage is in red, and the antibiotics that we saw were actually comparable to the municipal sewage. But then looking at time, the spike that we saw was actually the municipal sewage. First of all, clarithromycin was very high and then went down. And then we had spikes in erythromycin and also in sulfamethoxazole. And then that happened across all of our reactors, so we had triplicate for each condition. So this is a nice replicate across 9 reactors. So then the question is, well, how is that affecting the resistome? And so Connor did some hybrid assembly and co-assembly and binning to try to see who's carrying which ARG and which mobile genetic element, and then sorted at the top are ones that we knew were in the hospital sewage to begin with, and sorted at the bottom are ones we knew were in the municipal sewage to begin with. And then it turns out the ones that persisted, so the red ones, tended to be higher for genes that encoded sulfonamide resistance and macrolide resistance, the same antibiotics that we saw spike in the wastewater. So again, kind of a nice, example of what we expect, but is elusive to find of antibiotics and ARGs increasing in sewage. Now, trying to figure out what happened, so Connor did put together metagenome, assembled genomes, and look at the hosts of these ARGs that were being enriched. And interestingly, those bacterial hosts that were predicted did not explain the behavior of the ARGs. So again, that hints at horizontal gene transfer. And so to look at that deeper, he's recently developed a new tool called KROS. And in the KROS Assess workflow, you can basically look at in situ horizontal gene transfer And so part of it's dependent on this comparative and time series data and basically look at the evidence. So you have your antibiotic resistance gene, mobile genetic element, and finding the different contexts they're in. So in this gene sharing graph where the arrows are, you can see examples of where it's predicting sharing of ARGs across phyla. And then over here is the prevalence of those gene sharing events. Now, in terms of getting back to the idea of prioritizing our mitigation efforts, so ARGs are everywhere, but what do we care about? It's, well, if they're mobile or if they're in a pathogen. And so that's essentially what the meta-compare pipeline looks like. Now I'm calling it meta-compare 1.0 because later this week we'll have Monjura presenting on meta-compare 2.0, which basically differentiates the score where you're looking at acute human health resistome risk assessment, so escapee pathogens and the rank 1 ARGs that are of clinical concern, versus a broader score like, okay, we're generally concerned about mobility of ARGs among, pathogens, non-pathogens. So that's the ecological resistome risk score. But the upshot here, Monjura's surveyed many, many environments, and we broadly classified them as polluted and unpolluted. Guess what? ARGs are contaminants. Say no more. So back to the future, and back to another black screen. Metagenomics was a huge game changer that I could not have imagined when I started as an assistant professor in the early 2000s. And now here we are with AI, and it's gonna change everything. It is changing everything. And it's making my screen flash right now. When it does flash on, I wanna give a shout out. So this is not a paper from our group, it's one that recently came out, co-authored by David Graham and Yongguan Zhu. And so they used AI to harvest the literature and kind of see what are the gaps in knowledge in One Health. And I think this is a real shining example of what AI can do, because let's face it, no matter how smart we all think we are in this room, we can't do DNA sequence analysis in our heads. And we certainly can't read the tens of thousands of papers that are coming out exponentially. And so I think we need to embrace AI, but do so responsibly. And at the same time, recognize we still need to go in the lab. So that was one of the summary statements that David Graham made is, one of the things we learned from this endeavor is we need culture. We need metagenomics. And I'll add my own quote here. We need solid bench and field scale research, or AI is just going to produce a bunch of garbage in, garbage out. So don't worry, you all still have a job, and your job is to produce high quality research, starting with those research questions that you ask. And just a quick plug. So my heart goes to Dominic in organizing this conference. It's a lot of work. I'm going to be chairing a conference a year from now, fortunately a little bit smaller one than this, but would love to have you there. So it's a Gordon Research Conference on microbiology of the built environment. And we will have a session on AMR about my current passion, which is training the next generation of students. So I've been talking with you all at the coffee break and introducing you to students. And so yes, I do have my collaborators and I have 16 students here. And the idea is, what better training grounds? And so I hope that you all take the opportunity to talk, to learn from each other, because, you all are the future. And so part of this is we have a training grant that's funded by the National Science Foundation. And so the training grant supports the students to get cross-training in data sciences, environmental sciences, microbiology, and policy. right? Because ultimately, this is a very urgent problem that we need to solve now. And so we need to be directing the science towards developing these solutions. And hopefully we can get a solution to the AV system for the next speaker. But for now, I'd be glad to have any questions that you have.
Yes, thanks very much for your presentation. Very excellent. I'm Bin Li from Tsinghua University student campus. Actually, I did learn a lot from your presentation. Not only this time, but also back to 2013 in the EDAR7. Yeah, at that time I was a postdoc in Hong Kong U. So my question is that for the risk evaluation of AMR, especially in the environmental dimension, so what's your perspective for this research field? Okay, thanks.
Excellent question. I do want to make sure I'm clear that what I presented on MetaCompare, it's not a traditional risk assessment. So it doesn't get at that number. Like if somebody has this exposure, this is the likelihood of illness. And that's what we really need for regulatory purposes. But what MetaCompare can do is help you look at metagenomic data, compare environments, identify hotspots, and then prioritize where you take action.
Thanks.
I believe there's a question.
Yeah, you have a question.
Hi, Tim Vogel from the University of Lyon. I'm just wondering if you'd like to comment, if you have some feelings about the importance of co-selection in your systems and the possibility that they drive some of the things you're seeing.
That is an excellent question, and we didn't look at it here in what I showed you. But one of the things we were interested in is during the pandemic, a lot of people were washing their hands a lot and using antimicrobials all over the place, just swimming in it, right? So that's going down the drain. And so we did look at quaternary ammonium compounds, and then we looked at biocide resistance genes. So there's some interesting trends there as well. It's hard to say if co-selection was driving what we saw. I think we still have some sifting to do through the data. But I can say in other data sets, like often we're zoomed in on antibiotics and whether they're imposing selection pressure, and then it turns out it's really a heavy metal or something else that's really causing a co-selection event. So that's a good point.
Hi, this is Mohammad Islam from Washington State University. So it looks like the wastewater treatment plants are kind of breeding ground for ARGs or the AMR. So my question is that, in your opinion, what kind of treatments are kind of most effective to reducing the ARG transmission or proliferation of ARGs, and whether those treatments are still exist, or in which state are we this moment to have get those kind of treatment facilities?
Yeah. No, it's an excellent question. I mean, wastewater treatment plants are a two-edged sword, right? But I don't personally want to live in a world without wastewater treatment plants. And I think it's very encouraging, the results I showed and all the talks I'm seeing, that when they compare that influent sewage and activated sludge, ARGs plummet and mobile ARGs plummet. But the point is there's still some there at a microdiversity level, and there still can be important events like Joachim showed in his keynote that's where we need to zoom in on. And in the meantime, I think it's like any other emerging contaminant. You're looking at multiple barriers, right? So we're looking at water reuse systems where even we want to take that effluent and use it as a drinking water source. And so, that usually entails advanced oxidation and biofiltration and then some more disinfection. And what we see is the numbers get very, very, very low. And certainly lower than if you go and drink lake water or river water, right? So I think it's manageable if you have the resources, but just to put a plug for one of my students who's presenting a poster, actually a couple are presenting on what if the wastewater treatment plant isn't functioning very well? And one of those posters is in Mozambique, and one is in rural Virginia. So it can happen in our own backyard that, especially low-income communities, don't have the resources to maintain their wastewater treatment plant properly. And I think that's where we really need to worry.
So I'd like to introduce our next speaker, Dr. Stephanie Glaser, and the title of her presentation is Cultivation Strategies Have a Strong Impact on Studies of Environmental Resistant Antimicrobial Resistant Bacteria in the Environment. Welcome, Dr. Glaser.
Okay, thanks a lot for this invitation that I can give this talk here. I want to give a little bit a critical view on the opportunities to culture bacteria by using different cultivation strategies. And what I want to show you here is, or as the abilities to culture escape bacteria. We know all of them, they're occurring in hospitals, they're leading to diseases because they can't treat it anymore with antibiotics, they're getting more and more critical. With wastewaters, human and livestock wastewaters, we are continuously releasing them to the environment. And as you've already heard today, that in the wastewater treatment, on the one hand side, their abundance is getting reduced, but there's also a formation of multi-resistant strains, which is linked to the presence of nutrients and all contaminants in the wastewaters. When those bacteria are released into the environment, for example, plants are a good alternative host for them. They can grow in plants, and then they are coming back to our food chain. So, in my point of view, it's really important to culture those escape bacteria from the environments to see how many resistances they have accumulated and to assess the risks. So, but depending on their abundance and activity in the environment, it's somehow critical to culture them. So, I want to give you an overview, how can we culture bacteria? And no, it's basic knowledge, but I think it's important to think about the different ways to culture bacteria to evaluate at the end the outcome of studies. The first way is to culture them directly by a direct plating approach. That means you take your samples, you detach the bacteria from the samples, then you're making dilution series, and you put the bacteria on different kind of, most often here, selective media, which are differential, that you can get as good as possible your escape bacteria from the environmental samples. This sounds good, easy, but most of those media were developed for the clinical purpose. If you go to the environment, we have some problems. It's a quantitative approach, that's good, but a key problem is that the escape bacteria are very often low abundant, and they are very often under detection limits if we're using this direct plating approach. And they can quickly getting overgrown by a high abundance of other bacteria, especially if you look to wastewaters. And most often, escape bacteria are not really happy in the environment. They are dormant or having a viable but non-cultural state, or at least having a reduced activity. And if you put them directly on the agar media, most often they can't grow efficiently. So that causes problems with this kind of cultivation technique. That means that there's a low detection efficiency, partially, if you apply this approach. And this may lead to the underestimation of escape bacteria in the environment. And an alternative is now to use enrichment techniques. Here, the samples, including the bacteria, put in liquid broth, which can be either non-selective, most often buffer peptone water is used, or directly selective, including, for example, process for specific escape bacteria, and also partially directly antibiotics. And then after this pre-enrichment, most often overnight, we can streak the bacteria on the respective selective and differential agar media. This approach is not just qualitative, because we have already let the bacteria grow in the liquid broth. We can make it partially semi-quantitative if we apply here statistical techniques like the MPN technique. What we have now in those pre-enrichments, bacteria can grow more efficiently. So we have more bacteria we can detect on the agar plates, and partially the escape bacteria have time to recover, and then they can be plated on the media. But during the pre-enrichment, it can also happen that bacteria may exchange resistant genes, or that there's competition amongst those bacteria. So we have to be aware that there can also happen things which we would avoid if you could use the direct plating approach. I just want to show you some examples. I would like to start with the first time when I came to the cultivation of escape bacteria, when we looked in Germany to small German farms having biogas plants, and we compared the presence of escape bacteria in the input and output samples on those farms. And in this case, I want to show you some data for E. coli. Here you can just see some amplicon data, which shows how abundant, how diverse bacterial communities are, and how less abundant our escape bacteria are. E. coli is most often not detectable in those amplicon data in the biogas plant output samples. At that time, we started with a direct plating approach using a chrome agar for Eastern E. coli, and we were able to detect them in the manure samples, which were the input of the biogas plants. But we couldn't detect them in the output samples. That would have mean it was really an efficient treatment. But then we started to apply the selective pre-enrichments, and then we were able to detect a lot of ESBL E. coli in the output samples. You can see here the chrome agar for ESBL E. coli, which are pink on this medium. By direct plating, you have a complete overgrowth of the E. coli by different other bacteria, and they are so low abundant that there were just maybe 1 or 2 colonies on the agar plates. But after selective pre-enrichment, you can see it on the bottom, there was a high abundance of those ESBL E. coli. So we need the pre-enrichment here to get really a sufficient result to see the risk that still E. coli are released from the treated manure into the environment, and then come back to our food chain, like colonizing plants and so on. But what happens during the pre-enrichment? And that we studied here in a quite simple experiment. We took a manure sample, and this manure sample was once direct plated on TBX agar, another selective medium for E. coli, then TBX agar with cephalosporin to get just ESBL E. coli. And in parallel, we took the selective pre-enrichment cultivation, where we put the manure sample in the easy pros, including the antibiotic too, to get also the ESBL E. coli. We took from all of the 3 platings and streakings 40 isolates, and then we compared the isolates. We just look first to the presence of ESBL genes, and if you don't have a selection agent, you mainly got non-ESBL E. coli, just very few carried and resistant genes. But when we applied the direct plating and selective pre-enrichment, all of the isolates carried CTXM genes in our case here. Then we compared the isolates by using a very old fingerprinting technique, the box PCR. You can see it on the right side. That are the box patterns, always from the 40 isolates coming from the different pre-enrichment. And what we found, as expected, if you just plated E. coli, we found a high diversity of E. coli strains in the sample. When we applied the selective cultivations by direct plating and the pre-enrichment, we found only very few different ESBL E. coli in the samples. There was always one predominant genotype based on the box patterns. But when we looked more detailed, the genotype was different. The genotype in the direct plating approach, there's a pointer. In the direct plating approach, here, that's not working, GT1 was not present after selective pre-enrichment. Vice versa, there was just one strain which belonged to genotype 2, which got very abundant then after selective pre-enrichment. Currently, we look into the genomes that is done by Nicole Rika, who had talks this morning, and we found that the genotype 2 belongs to an ST type, which is already known to be fitter than others. That is a reason why it grew more predominantly during the pre-enrichment. But when we compared the one strain of genotype 2, which you obtained by direct plating, we saw it had just 1 resistant genes. But the other ones had a lot of resistant genes. That means it looks like that during selective pre-enrichment, we also have an accumulation of resistant genes. That are just preliminary data, but here I just want to point out that be critical which kind of cultivation techniques you're using. Another example, we incubated lettuce samples, or we irrigated lettuce samples with treated and untreated wastewater. And here we used again selective and non-selective pre-enrichment techniques to culture the bacteria here from the untreated, the lettuce samples at the bottom, irrigated with untreated wastewater. From leaves and roots, we were able to culture them. Again, we applied genomic fingerprinting, and here we found out that just with a selective pre-enrichment, we could obtain a multi-resistant strain which was dominantly colonizing leaf plants. When we applied the non-selective pre-enrichment, we have completely overseen that strain. So again, it's critical, check which kind of techniques you're using to culture the bacteria. Another example, one of my students is culturing Acinetobacter baumannii. Here he's using a chrome agar medium, Acinetobacter, where Acinetobacter forms red colonies. A problem, Pseudomonas has the same kind of colony experience, so you can't quantify with this medium. But you can culture Acinetobacter from these different kind of environments. Here he looked to wastewater treatment plants and biogas plants again. And he played a lot around, and the efficiency to culture Acinetobacter got more and more better as higher the temperature was. So he was able to culture Acinetobacter baumannii, especially for all those kind of samples. But here I want to mention one point. Take care. You can enhance your efficiency if you increase the temperature for incubation. But a problem is you also have to take care that you may oversee bacteria which cannot grow under high temperatures. He found one Acinetobacter strain, it belongs to the species Acinetobacter bohemicus, which came originally from soil and water samples from the Czech Republic, which was non-human impacted. And the strains we found in the wastewater treatment plants have already accumulated a lot of heavy metal resistant genes and mobile genetic elements. So this bacterium is also important, maybe for the transmission of resistant genes. So it has to be taken care that you don't forget to play around with temperatures of incubation to get also environmental strains which may be important for the transmission of resistant genes in the environment. I just want to point out here in our AMRS consortium, we have established for a lot of different escape bacteria direct plating and selective pre-enrichment techniques. Heike Schmidt has a poster, and Nicole Rieker has a poster where those results of this research is shown today and tomorrow. So in summary, please take care which cultivation techniques you are using. It's really important to study the respective samples. Cultivation conditions can strongly impact the outcome of your studies. So, and with that, I want to thank our cooperation partners, where we have already applied all those methods for our analysis.
I think it's a great talk. It's about the direct streak from this kind of antibiotic-resistant bacteria in the wastewater and escape bacteria, which can cause the direct threat to human being and the pathogen. I think with more and more effort on the isolation and metagenomic analysis about this kind of escape gene, escape bacteria, pathogens, then we may understand more about AMR in the environment and also in human and animal sector. I think absolutely you are right. The cultivation condition may have huge impact about result. And you suggest that we need to do some pre-culture and using some conditions. And I wonder, this kind of pre-culture process may have some bias because that the quantitative information may be different from the original sample. What could be your comment on this and any suggestion for that? Thank you.
Yes, there is a bias, but partially, if you don't use this abbreviated selection, you can't culture them at all. So you always have to use, in the best way, both techniques, and then compare the results.
Thank you. This is Luis Motoro from the University of California, Irvine. So the chrome agar was developed for urinary tract infections, where the bacteria is already in liquid. However, most of the bacteria that exist in a solid surface, such as feces, are in biofilm form. So they are trapped in matrix. And that's why it's not easy to get them out. So I wonder if you have studied this matrix of bacteria, or if you have reviewed the literature as to what other bacteria they are associated with, and that's why we are not able to get them in agar, immediately after coming from the solid.
Yeah, I think So you mean the bacteria are attached to biofilms, and so you can't directly culture
because
them?
they're matrices,
Yes,
and they are
for
very
sure.
solid matrix.
Yeah,
It's very hard to get
especially,
lose them.
and I think that's a problem if you have bacteria, for example, attached to plants, because at plants they form the same kind of biofilms. And in this case, however, we'll have a bias with a pre-selection, pre-incubation. We have to apply those techniques, because otherwise we would oversee them because they're still sticking to the plants, and we would not have them in the liquid to make this dilution plating. So that's the reason why I think we need both approaches.
Thank you.
It's my pleasure to introduce our next speaker. It's Dr. Donald Morrison, and the title of his presentation is Bathing Water-Based Epidemiology, 4 Years AMRsurveillance at Scottish Bathing Water Sites 2018 to 2023.
So my background is in human medicine, human microbiology, and for 20 years I've been looking for AMR in hospitals and in the community, and surveillance methods have been set up for a number of these. And the prevalence, what is the prevalence of AMR in samples, is kind of the big figure that we've been looking for. For the last 10 years, I've been working in the environment, looking in the environment and looking for AMR in the environment. This was a study that was done in collaboration with SEPA. SEPA are the Scottish Environmental Protection Agency, who, as well as many other things they do, they look at bathing water quality. And this was a study that they have done the hard work for over the last 6 years, and we have 4 years of data of AMR in the environment. And the kind of take-home message from this study is that doing bathing water-based epidemiology is cost-effective. It didn't cost them very much to implement this as part of their bathing water quality. The other take-home message was that we are able to screen large numbers of the target organism, in this case E. coli and intestinal enterococci. So vast numbers of organisms are screened throughout the year. And the final take home is that we think that there is correlation between the data we get in bathing water to what you would get in other environmental surveillance schemes. So this is the bathing water quality data that SEPA produce every year. They have, on the whole, bathing waters in Scotland, and there are over 80 of them, 89 in 2023. On the whole, bathing waters in Scotland are of good quality, with 85% of them either being of excellent or of good quality. So what SEPA did, and it happened in response to a student I had, a project student, over a summer period, and she was looking at AMR in bathing water. After that project, SEPA incorporated her method into their technique. So they sample 89, it was in 2003, bathing waters. They do it from June to September, and they take around up to 18 samples from each bathing water every year, and they're looking for enterococci and E. coli. So their integration of AMR surveillance was very simple. Rather than taking a 1 ml or a 10 ml sample, they used 100 mLs for looking for AMR, and they used 2 extra plates in their sample. They used it was a chromogenic Geiger TPX, as we've heard of before, and using 4 milligrams per litre cefotaxime and 8 milligrams per litre of vancomycin, based on clinical breakpoints, not ECOF, and they use a membrane filtration method. So what we find, this is a tool, that data visualization tool, which SEPA have put up on the website for anybody to look at. And just quickly, you can see that in the red is where we found AMR at bathing water sites, and the green is where we didn't find them. And I'll go through a couple of these maps shortly. But this is what we found. There are AMR at many bathing water sites throughout the year in Scotland. To look at the data, we asked 3 questions, and these are the ones that are asked of surveillance systems in general, and this has been taken from Huber's paper at the bottom there. So basically we're looking for prevalence. What is the prevalence? And we're looking for population level prevalence. And as I've said, the numbers that you can screen using bathing water samples is very large. We are screening around 200,000 E. coli every year. And we're asking what's the proportion of resistance within that population. Then we ask the question, what's the risk of evolution? Can we see over time an increase in resistance in these populations? And thirdly, what's the risk of transmission of these resistant bacteria to water users? And to do that, we're using the markers of the total number of ARB and, of course, fecal pollution itself is a marker. So looking at the population resistant prevalence, what did we find? This is the data, and as you can see, large numbers, 155,000 E. coli screened in the year, and on average 200,000. So of that 200,000 E. coli that we screened each year, this gives you the prevalence, the proportion of these organisms given as a percentage figure. So we got 0.3% in 2018, a higher level in 2019, and many of the figures you'll see are higher in 2019, and then equivalent levels in 22 and 23. No data in 20 and 21 because they did bathing water sampling, but not for AMR due to the pandemic. Enterococcal figures on the far side, again, high numbers screened of 106,000 enterococci. There were 1.59% of them were resistant. As you can see, the numbers are low. However, note that the enterococcal numbers are much higher. There's about 3 to 4 times the level of enterococca of vancomycin resistant to cefotaxime resistant in E. coli. This is just to compare the data with other surveillance systems. So in Scotland, we have surveillance of clinical isolates, bacteremic isolates, E. coli for cefotaxime resistance, and the levels are much higher, as you can see, from 8.2%. But again, and this is what I was saying, that there's some correlation between the data we get in the bathing water with what is found in clinical surveillance. The number of samples surveyed for the clinical data in Scotland was 4,000 E. coli compared with 200,000 E. coli in the bathing water. And the picture is that there has been little change in the 6 years in clinical isolates and relatively little change in the bathing water isolates. The data for England and Wales are next to them. For enterococci, again, we have only done these in the last 2 years. But again, we have a particular problem with Enterococcus faecium, vancomycin-resistant Enterococcus faecium in Scotland. It's one of the highest figures in Europe at around 40%, double the percentage found in England and Wales. We don't know why that is. But interestingly, the figures for the bathing water proportions of resistance are equivalent to what you find in the cefotaxime. So cefotaxime resistance in the clinic and in the bathing water are equivalent. 3 to 4 times more found in the clinic and 3 to 4 times more found in bathing water. So although the percentage is less because you have dilution and inactivation of the E. coli and enterococci as they make their way to the bathing water. The second question we asked was, what's the risk of ARP evolution? Did we see any trends in the evolution over these years? Well, we saw with the proportion, one of the markers of increase in or decrease in antimicrobial resistance, we saw there was little change. So we also looked at antimicrobial susceptibility data, and we tested a number of different antibiotics, and it's a mixed picture. It's for 3 years, this data, 21 to 23, but on the whole we found 5 antibiotics, these first 5, which are showing some decrease over the 3 years, and in the other 5 antibiotics along here, there was some increase. So a mixed picture, only one isolate with carbapenem resistance. The final question we asked was, can we assess whether there is risk of transmission with this data? And here we're using the absolute numbers of ARB and fecal pollution. So here's the map I showed before, and you can see, as we've seen before, there are a large number of our bathing waters which carry or which ARB have been detected in. This is for cefotaxime resistance data. However, the map overestimates what we're finding. Maybe one of these red symbols, it might have been just one sample of the 19 that were positive, so we need to drill down a wee bit. The size of the rectangle, the diamond here indicates the number of samples that were positive. And you can see some of the inland ones were very high, but again, the Firth of Forth over here near Edinburgh and the southwest of Scotland have got particularly high numbers. In the last 2 years, 2022-23, we've also been looking for VRE, and they are shown, when we find both of them, they're shown with the large squares. But again, we need to drill down for this data to see what we're finding. If we look at the samples, so again, a large number of samples are taken. But of these samples, only a small number of them are positive. So 30 seconds left. Okay, so around 9% of the samples are positive. And from this data, you can see from by month in each year what we're finding. And what we find is that it is periodic. You may get one beach which is positive in one sample in 1 month and then not in the other months. So the conclusions we've come to is that bathing water epidemiology is cost effective. It screens a large number of isolates, and it seems to mirror what we find in other surveillance. The 0.5% is what I find looking at effluent from wastewater treatment plants. No trend in the prevalence, little evidence of evolution, and the risk of transmission seems to be low. Thank you.
Yeah, Mohammed Islam from Washington State University. What's the bathing water, by the way? I'm not quite familiar with that. Is it surface water or municipal water? What kind of water is it?
Okay, I should have made that clear. And in Scotland, all apart from 3, so we have 89 designated bathing waters, and only 3 of them are fresh water. So they are marine saltwater bathing waters. My apologies, that was an obvious thing to have told you.
Kathy Curlow, Canadian Food Inspection Agency. I'm curious if you're keeping the isolates that you're collecting for comparison to clinical, so that we can make those sort of connections by genome sequencing or other methods.
So are we doing further analysis on them? So we haven't so far, but the intention is that these will be, there's another lab in Edinburgh where they're going to look at do some whole genome sequencing to get some further data. Yeah, so we are going to do more analysis on them. And the one problem, of course, is that the one issue is that the strains we find in the bathing water, are they a complete subpopulation of what we find anywhere else? And that's what that data would show.
Okay, Great talk. Ishi Keenum from Michigan Tech University. And I'm just getting interested in this because I've moved to a place with lots of lakes. So I was wondering, are you seeing that you get AMR bacteria always when you get upticks in regular beach monitoring of bacteria? Or is there any kind of, when I talk to public health officials, they're like, okay, well, do we need to do this at every site? Or is it maybe we can, if we're seeing upticks in regular E. coli, we can then start doing AMR specific?
So I think so. The data shows that when there's high AMR or ARB, there is also high fecal counts. So the two, on the whole, not always, but on the whole, they link together.
So we have time for one more question.
Heike Schmidt from the National Institute for Public Health and the Environment in the Netherlands. Very nice talk. Actually, maybe two questions. One of them is, did you look out for risk factors that might explain in which locations, for example, wastewater treatment plants or animal runoff would cause some of this contamination? And the second is, how are your results received by the general public or by the responsible institution that is responsible for bathing water quality?
So we haven't looked further into the data, but that's obviously the next step for us. And who sees this data? So the data on these nice maps, they're available to the public. And in Scotland as well, every time, even with the fecal indicators, they have signs on the beaches where they indicate whether it's a high or a low, whether you should or not bathe.
So the next speaker is Dr. Mohammed Islam from the Washington State University, and he will talk about genomic and metagenomic insights into antimicrobial resistance in human, poultry, and wastewater environments in urban and rural Bangladesh.
Thank you very much for your kind introduction, and I'm absolutely delighted to address this distinguished audience. And today I'll be talking about the genomic and metagenomic insights into AMR in a kind of One Health approach that we have taken, and the context will be in a developing country setting in Bangladesh. So yeah, I'll start with this nice schematic diagram. As you all know, that the AMR transmission pathways in the community is really complex. As you can see in this figure, there are many different drivers and many different pathways that AMR can be transmitted to the community. So one of the important aspects is that the food-producing animals, especially those which are basically raised on antimicrobial prophylaxis, could serve as a kind of reservoir for AMR, antimicrobial resistant bacteria and genes, that we all know. And therefore, the exposure to these animals and their proximal environments could be one of the risk factors for transmission of AMR to the humans, especially in those settings where antibiotics are being indiscriminately used in food production, and that is particularly more frequent in the developing countries. But there have been very few studies in the One Health approach to see the risk and the study. And despite the fact that the LMIC countries are disproportionately being impacted by the consequences of AMR in terms of the disease morbidity, mortality, and economic consequences. So a few years back, we have done a study in Bangladesh. It's a kind of observational One Health study where one of our main objectives was to see the impact of human exposure to these food-producing animals, especially the poultry, on the colonization with antibiotic-resistant organisms, in this case, the extended spectrum beta-lactamase producing E. coli among humans in Bangladesh. And in that study, what we have done, we selected 3 different ecological settings where people usually have frequent exposure to poultry. One of these is the small or medium scale poultry farm, and the other one is the live bird markets in urban areas where usually people can buy the live birds and they usually slaughter it, process it, and deliver it to the customers. And then the other one is the rural areas where people have, the backyard chicken. And in all these 3 areas, we selected people who are having the frequent exposure to poultry. And at the same time, we also selected people who are living in the same areas, in the, the same age group, but they do not have the frequent exposure to chicken. And we collected the stool samples from those peoples, and then also collected fecal samples and the chicken cloaca samples from the chicken, and the environmental samples from proximal areas, including like the wastewater, drinking water, surface water, river ponds, and then solid waste. And we tested all these samples for ESBL producing E. coli. What we have found is basically, between the people who are having frequent exposure and low exposure to poultry, we didn't see a lot of difference, but, on an average, more than 65% of the people were colonized by ESBL producing E. coli. Although you can see some slight differences, but those differences was not statistically significant. Even if you look into the, the level of colonization, those were also not significantly different between each other. So, so this basically highlights that there might be something different other than the exposure to poultry. Probably there are some common factors which are involved in this high level of colonization with these organisms. And when we looked at the ESBL EC positivity among the other samples, we found that the wastewater samples basically were predominantly positive for ESBL E. coli compared to the poultry and the human samples. And in case of poultry samples, we found that the poultry from the urban live markets, were predominantly positive for ESBL E. coli compared to the poultry samples from the rural areas. But most interesting thing, if you look at the colonization in humans, the human had a higher prevalence with ESBL EC compared to the poultry, which are sometimes, reared with antibiotic prophylaxis. So now the questions that we have asked here: do human and poultry share the same antimicrobial resistome or microbiome? How about the antibiotic resistant organisms like the ESBL EC? Do they share the same organisms? And how about the proximal environments? Those do this environment play any role in transmitting the ESBL EC or resistome from poultry to humans or vice versa? And so to address this question, we did whole genome sequencing of 117 isolates, the ESBL E. coli isolates, and shotgun metagenomic sequencing of 40 samples collected from all 3 sources, representing both urban and rural areas in Bangladesh. And what we have found is the whole genome sequencing data, the maximum likelihood phylogenetic tree based on the SNP distances using the whole genome sequence data. And as you can see here, these are the blue and green highlights representing these clonal overlaps based on less than 22 SNPs differences. And we got quite a few clonal overlaps, but in most of the cases, those clonal overlaps were the isolates within, the same sample types, like, these isolates from the environment to environment, isolates from poultry to poultry, human to human. But we didn't see a lot of, the clonal overlaps between different sources. I have elaborated here some of these examples, excluding the isolates from solo single sources like environment to environment. Here I'm presenting these isolates from human with high exposure to poultry and then the environment. But in most of the cases, we found that the human isolates clonally overlap with environmental isolates, and then poultry isolates clonally overlapped with environmental isolates, but we did not see any evidence of clonal sharing between human and poultry, human or chicken. So then we looked into the ARG diversities among the isolates, and we found that the environmental isolates had the significant more diversity compared to the human isolates. When we looked at into the diversity according to the study sites, we found that the isolates from the wet markets, urban markets, had the highest diversity compared to the significantly higher diversity compared to the poultry farms and rural households. we also wanted to look specifically into the wastewater, and we found that the wastewater samples from the urban markets had the significantly higher diversity compared to the wastewater from poultry farms and the rural households. So then we also looked into whether there are plasmids carrying the antibiotic resistant genes shared between the isolates, and we found a total of 7 cases where we found the same plasmids carrying the antibiotic resistant genes are shared between the isolates. But interestingly, we did not find any of the isolates from the human that was, the shared with the environmental isolates, or human with human, chicken with chicken. We found that the chicken isolates shared the same plasmids with the environmental isolates, and then among the environmental isolates, the wastewater was the predominant one, and those wastewater were from the urban live bird markets. And this was also corroborated with the metagenomic sequencing data that we found. As you can see here, the wastewater samples from the urban markets had the highest abundance of ARGs compared to the wastewater from the rural areas. And in terms of the ARG diversity, we also found that the ARG diversity in the wastewater, especially from the urban live markets, had a higher diversity compared to the humans and the poultry. And so it looks like that the humans and the poultry and the environments, they were kind of distinct in terms of their registome, which was also, we found when we did this principal coordinate analysis of the registome. As you can see here, the human samples, the registome of the human samples and the poultry samples are environmental samples. All are, they clustered but dispersed separately based on the canonical analysis of the principal coordinates. Then when we looked at the microbiome composition to some extent, we found they were also quite distinct. In case of humans, we found that the human samples were basically dominated by the Prevetola. There was a significant higher abundance of Prevetola, which was scarcely found in poultry and wastewater samples. On the other hand, in poultry samples, we found quite a few abundance of the Bacteroides, which was scarcely present in humans and the wastewater. But in case of wastewater, we found some of the environmental isolates quite abundant, like the Aeromonas, Comomonas, and Acinetobacter. So those were not found in the human and the poultry samples. So when you go back to the questions that we originally asked in these settings, basically we did not find any, the sharing of the registome and the microbiome between the human and poultry, and even, we did not see any sharing of the isolates, the ESBL producing E. coli. However, the proximal environments could play a role in transmitting the ESBL E. coli as well as the antibiotic-resistant genes between these two sources. And therefore, I think reducing the exposure to the contaminated environments could be a potential intervention strategies to control AMR in these settings, and this can be done by improving the water sanitation and hygiene conditions in that particular settings. So yeah, I will be stopping here, and to acknowledging my team at the Washington State University, my collaborators at ICDDRB in Bangladesh, where we have done this study, and my collaborators at Loughborough, UK, and the University of Nottingham in UK, and the funding agencies NERC, UK, Washington State, ICDDRB, and Loughborough. Thank you so much, and I'll take any questions if you have.
Thank you so much for the presentation. I'm Mohammad Usman Zahir from Food and Agriculture Organization of the United Nations, Regional Office in Asia and the Pacific. I found your presentation to be very interesting, and given it's One Health approach that you have taken, I just wanted to ask a couple of questions around what are some of the practical challenges that you think countries can face when they are starting to have monitoring of AMR in the animal production environment. The reason I'm asking is, as per the FAO's mandate, we are supporting the member countries in strengthening their AMR surveillance in agri-food sectors, including the food production environment. And we are at this stage developing a guide for countries that they can take it forward to conduct monitoring of AMR in the food production environment, and we are developing a guide. So I think your input to that will be very helpful. And of course, we are also having a workshop on Wednesday. So if you can join that, that would also be very grateful. The workshop number 1, which is on the environmental prioritization of the research needs as well as the guidelines. Thank you.
Thank you so much. That's a very good question. I think currently one of the constraints is the laboratory capacity, especially in the food side. There have been quite a few development in the human side, but in the veterinary side and then the environmental sides, there have been a significant gap. And so the development of the laboratory capacity would be something that needs to be considered. And the other thing is that sustainability. Even I think if you start with something, if you make something, then to make it sustainable, I mean you can invest, but then after the initial investment, to sustain that capacity is a big challenge. If you work with the government system, the people who you will be training today, those guys will be transferred to other places. So to keep them, working in the sector is a very difficult task, and I think it's an ongoing challenge. I don't know how to solve that, but in terms of like the laboratory capacity, skilled manpower, and then sustainability, those are the three things that needs to be considered.
So question. So I'm Luis Motabravo from the University of California, Irvine. So my question is regarding your last slide, where you showed that poultry, humans, and the environment have a very different composition of bacteria. So what that is telling me, and that shows in many other studies, is that each of these environments are selecting for different type of bacteria. So if that is the case, why are we expecting to have the same proportion of antibiotic-resistant genes when they are actually selecting for completely different type of bacteria? I mean, they have different temperatures, different pH, nutrients, and so on. So each of these are very selective environments. So why are we keep testing the idea that they should have the same proportion of genes when they are completely different environments? What are your thoughts about that?
That's a very good question, and that's my question too. So it's basically my understanding is that this is a kind of small scale study. The number of samples was not that much, I would say. But I think there is definitely a gap in those particular settings to have this kind of study within using the One Health approach and then integrate with the genomics and metagenomics. But there are the studies out there, bigger studies, and those studies have found that probably these antibiotic resistant organisms to some extent could be niche specific. And I don't know, how much transmission, clonal expansion is going on, but definitely the mobile genetic elements and horizontal gene transmission could be something to look at. But this is a valid question, and we need to get those, the answers to these questions.